I would also like to start by thanking Dr. Sharat, Dr. Lermark for generous introduction. And I would like to be unrestrained in my gratitude to Crafoord family, to Crafoord Foundation and Crafoord Prize Committee for the tremendous honor. I am particularly honored to be recognized jointly with Dr. Shimon Sakaguchi and Dr. Fred Ramsdell today. I'm also delighted to see many people in this audience not being scientists, but being students, high school students, undergraduate students. And if I have time towards the end of my talk, I will elaborate a little bit more on this topic. In today's lecture, I would like to discuss our studies of fascinating biology of regulatory T cells in a broader biological context of immunity. I would like to make first 3 points. First, that immune system of multicellular organisms evolved to maintain integrity of multicellular organisms, in particular upon infection and injury. Inflammation is an adaptive reaction aimed at restoring tissue function and protection of multicellular hosts from infection and promoting tissue repair and restoring homeostatic set points, allowing organs and organisms to operate. The benefits of immune response associated inflammation are constrained, limited by potential impairment or loss of tissue function. So it's essentially a deal, a trade-off, acquisition of the ability to mount very effective immune response and the penalty that results in impairment of function in disease, such as autoimmune disease discussed by Shimon, that results from this acquisition. Evolutionary ancient innate immunity, innate immune system, features a set of sensors that were selected in the evolution. They are hardwired sensors that are recognizing microorganism-specific molecular shapes or patterns, called by Charlie Janeway pathogen-associated microbial patterns. For example, there is a particular glycolipid found in bacterial cell wall that is not found in multicellular organism, in humans or in also animals down the line. In addition, the sensors can sense perturbation of physiological parameters of the host. That is, when a pathogen, virus or bacteria can perturb the function of the cell. So there are sensors that sense this perturbation, not necessarily a gene product of virus or bacteria, but the fact that the cell doesn't work. For example, certain ions are increasing in concentration inside the cell and then triggers activation of the immune system. Another feature of innate immune system is that this individual cells and prototypical cell of the innate immune system as a phagocyte co-express multiple receptors by a single cell. And this is all proven and all selected, and that allows innate immune system to recognize broad classes of pathogens. Unlike innate immune system, adaptive immune system, and its principal cells, T lymphocytes and B lymphocytes, or T cells and B cells already introduced to you by Shimon, feature a vast array of receptors, essentially unlimited diversity of receptors and sensors capable of recognizing any molecular structure. These receptors are generated somatically during differentiation of lymphocytes. And they are generated in a combinatorial manner by splicing distinct genic fragments into functional gene that encodes receptor. So this diversity is generated in anticipation of future encounter with pathogen. Essentially, any structure can be recognized. And this is the answer of multicellular organism to high rate of mutations and high diversity that is found in microbial world. So that's the schematic, and this is taken from the Janeway book, the famous textbook written by Charles Janeway, who was actually a great friend and fan of Sweden and knows a number of, or knew a number of people in Lund and elsewhere in Sweden. So what is happening during differentiation or development of lymphocytes, genes that encode T and B cell antigen receptors, and this is immunoglobulin receptor in its cell-bound form in B cells, and T cell receptor, already introduced to you by Shimon in T cells, rearranged. They are expressed in a clonal manner. And these colors represent individual flavors of these receptors, so that one lymphocyte, in extreme case, would express one unique receptor. So these receptors are subject to selection that also Shimon mentioned. That is that self-reactive lymphocytes, developing lymphocytes and mature lymphocytes that express self-reactive receptors are being eliminated. This has been recognized early on by Macfarlane Burnett, who came up with the clonal selection theory and predicted elimination of self-reactive lymphocytes. So even before, sort of basically, Macfarlane Burnett, a virologist who came up with clonal selection theory essentially on a theoretical, hypothetical basis, recognized the danger of self-reactivity that would come from this generation of clonal structure. So then this process would result in elimination of the majority of self-reactive cells. And then lymphocytes purged of self-reactive cells enter in the periphery where encounter with a pathogen, foreign antigen would result in clonal expansion and differentiation of the cells into so-called effector cells that are capable of eliminating antigen. So while immunoglobulin receptors or B cells can see any structure and bind antigens as they are, antigen receptors of T cells recognize protein breakdown products, peptides, short fragments of proteins that result from protein breakdown bound to products of major histocompatibility gene complex, or MHC. So this is this groove, peptide binding groove. It's like a baseball glove that holds on to peptide fragment, protein fragment, and this is T cell receptor recognizing peptide MHC complex. So I got this slide, and perhaps you can better see it if I turn on the movie. You can see that these are the variable regions, this hypervariable regions of T cell receptor, these little loops that are interacting with both peptides, and this is a ribbon diagram of real crystal structure solved by Chris Garcia. They are recognizing with both alpha helices, these parts of MHC molecule on the peptide, and this is projection of the peptide. So MHC molecules are highly polymorphic. So many people here in this audience, so it will be difficult to find complete MHC match that will, if we do the MHC typing among people present here. And another point that I would like to make is that T cell receptor intrinsically is biased towards seeing MHC molecules, and that was suggested first by David Rollet and shown by Chris Garcia through crystallography studies. So in addition to being biased to MHC recognition, the selection process of T cells in the thymus, it's sort of school where T cells are being educated, is selecting T cells on the basis of weak reactivity with self-peptide MHC complexes. So that means that T cell recognition, its in nature, is self-referential. And this is sort of term was coined by Charlie Janeway. So this self-referential nature of T cell recognition presents a major problem of self-non-self discrimination. So one way, but imperfect way, to address this issue is the requirement for second signal that Shimon, Dr. Sakaguchi mentioned to you. And this is the example of requirement of two signals for activation of lymphocytes. Here you see an antigen-presenting cell. It's one of the cells of the innate immune system, very important in the immune system, called dendritic cell, recognizing bacteria, and it recognizes this microbial pattern shown in blue through toll-like receptors, innate immune sensor, that senses, for example, a glycolipid of the bacterial wall. So this signal allows for efficient phagocytosis of bacteria, where bacterial antigen, shown here in red, is chopped and presented by MHC class II molecules to T cells. And there are two classes of MHC molecules, class I and class II. At the same time, the signal induces gene expression that results in expression of co-stimulatory molecules, this essential, foolproof second signal, and cytokines. And together, these three signals promote activation of T cells. So the signal tells T cell that the antigen is legitimate and has to induce a protective, potent immune response. So this is an example, in a way, of a positive feed-forward regulation, where you can see that the signal through toll-like receptor would result in changes in gene expression, in upregulation of co-stimulatory molecules and stimulation of T cells. In addition to positive feed-forward, there are also positive feedback regulation when activation of one unit, one component of this regulatory unit would induce activation of another component of regulatory unit, that it would feed back and promote activation. So this feed-forward regulation, along with negative feed-forward and negative feedback regulation, are permeating all biology. This type of regulation can be found across the different fields of biology. And pretty much, these principles of regulation, they of course are employed outside biology. And these principles of regulation are sufficient to provide control and balance in operation of different biological systems across different organisms. But I would like to argue that it's insufficient for the adaptive immune system. And that is because effector CD4 T cells in particular act foremost as amplifiers of innate and adaptive immune responses that employ both positive feed-forward and positive feedback regulation. As shown in this example, activation of phagocyte, and MF in Greek stands for macrophage. The cell takes up bacteria, is being activated, produces effectors of innate immune response. It activates T cells, and this activated T cell feedback and activate this macrophage even further and activate other macrophages, and the cells proliferate. So it's very powerful feed-forward loop. This amplification of immune responses by CD4 T cells combined with their self-reactivity, built-in reactivity with self, essentially, we think, makes it impossible for this conventional means of negative regulation to function and to functionally control these penalties associated with the emergence of the adaptive immune system. And that leads to a new principle of regulation, and that is, you can call it negative feedback regulation in trance. And as we already know from Dr. Sakaguchi's talk, this negative feedback regulation in trance is mediated by subset of CD4 T cells, now known as regulatory T cells, that express X chromosome-linked FOXP3 gene. This regulation is sort of unprecedented in biology, but there is one precedent. And this precedent can be found in the nervous system. And it's interesting and intriguing to think that the two major sensory and integrative system, immune system and nervous system, utilize the same principle, and essentially analogs of regulatory T cells in the nervous system are inhibitory interneurons. The neurons that are specializing in limiting activation, signals that are transduced by excitatory neurons. So what we now know is that regulatory T cells, or Treg cells, as you know, are principal regulators of inflammation in diverse settings. In autoimmunity, as described Shimon, in inflammatory disease and hypersensitivities, they regulate host commensal interactions at mucosal barriers and therefore mucosal barrier function. They limit responses to infection, inflammation associated with both acute and chronic infection, allogeneic conflicts, human-made allogeneic conflicts being transplantation, and allogeneic conflict that emerged in placental animals, pregnancy. They participate in tissue repair. Recent studies suggested they limit what you would call sterile or metabolic inflammation, such as atherosclerosis, type 2 diabetes, and other disorders, and are prominently featured in cancer. What I would like to do is just to provide few facets of our work and illustrate some of these features of regulatory T cells. And what I would like to mention is just to see how profound this effect is. Already Dr. Sakaguchi and Dr. Ramsdell will already described IPEX syndrome, but I would like to point out the severity of the disease and the breadth of the disease affecting different tissues in boys affected by IPEX syndrome. And these are FOXP3 knockout mice generated in the lab by former graduate student Jason Fontino, and these mice also developed the wide spectrum of lesions in diverse tissues and organs, and they die by 4 weeks of age. When we generated this data and looked at these mice, we had difficulty ascertaining defective development of regulatory T cells because mice are sick. So what Jason Fontino decided to do is experiment called mixed bone marrow chimera. When he transferred bone marrow hematopoietic stem cells, and you know that all cells of the blood are originating from a single stem cell, transferred hematopoietic stem cells from wild-type mice, and from wild-type mice in red, and from mice lacking FOXP3 gene that he made in black. And he found that in this mixed bone marrow chimeras, and recipients were irradiated to eliminate the host hematopoietic cells, regulatory T cells were derived only from FOXP3 sufficient, but not from FOXP3 deficient bone marrow. And this retroviral transfer experiment of FOXP3 gene Dr. Sakaguchi already described. So this suggested that FOXP3 is required for differentiation of regulatory T cells in normal mice. And subsequent experiments by Jason showed that lack of regulatory T cells is fully responsible for all manifestations of the disease observed in FOXP3 deficient mice and by extension in humans. And he came up with 4 arguments that I would like to list to you. And the reason for us to doubt this was because most of the so-called transcription factors, gene products that control expression of other genes, normally utilized by multiple cell types and different combinations in development for their development and function. So what Jason did, he inserted sequence of fluorescent protein that was isolated initially from jellyfish, green fluorescent protein, into FOXP3 gene locus, so that the functional FOXP3 protein was expressed, but cells were marked with green fluorescent protein. And to our surprise, he found that FOXP3 was exclusively expressed in a subset of T cells with suppressor function and nowhere else. Since he found that the gene was expressed in T cells only, he decided to do another genetic experiment and eliminate FOXP3 gene not in all cells as he originally did, but only in T cells. And you can do genetic tricks that would allow for elimination of genes in a cell type specific manner. And he found that phenotype of mice, disease in mice where FOXP3 gene was lacking only in T cells, was identical to mice where FOXP3 gene was lacking in all cells. Further studies by Jason and former postdoctoral fellow in the lab, Chi showed that FOXP3 gene was fully dispensable for all these processes that Dr. Sakaguchi described to you, this cell-intrinsic elimination of self-reactive clones in the thymus, energy induction in a cell-intrinsic manner, right, or activation of cytokines in non-regulatory T cells. So it seemed that the function of FOXP3 gene and expression was limited to regulatory T cells. And furthermore, Jason was able to rescue the deficient mice by transferring small number of regulatory T cells in newborn mice, very much like Dr. Sakaguchi in early experiments was able to rescue day 3 thymectomized mice by transferring T cells. So taking advantage of this reporter gene, Jason was able to show that FOXP3 expression is dependent upon T cell receptor signaling and interleukin-2 receptor signaling. By breeding reporter mice, expressing FOXP3 reporter, this fluorescent protein, and mice lacking either interleukin-2 receptor subunits, one or another interleukin-2 receptor subunits described to you, or mice lacking all MHC molecules, and T cell receptor will not be able to recognize these MHC molecules because they were lacking. And in this mice, there was no expression of FOXP3. So in addition, Wanjun Chen from NIH showed that some naive T cells can upregulate FOXP3 gene and give rise to so-called extrathymic regulatory T cells when they're activated in particular manner. So that puzzled us and raised the question, what is the feature of T cell receptor that allows for FOXP3 gene to be expressed? So are the T cell receptors that are expressed by this protective anti-inflammatory regulatory T cells different from receptors expressed on conventional cells? And this was addressed early on by former postdoctoral fellow in the lab, Chi, who analyzed these T cell receptor repertoires by sequencing DNA sequence that codes these receptors. And he found that regulatory T cells express distinct, highly diverse repertoires of T cell receptor that are self-reactive, that have increased self-reactivity. And this repertoire is partially overlapping with receptors found in non-regulatory T cells. Another puzzling thing, and just follow me, in mice lacking FOXP3 gene and lacking regulatory T cells, she found that activated effector T cells frequently utilize T cell receptors that are normally found on regulatory T cells in normal mice. Is that clear? Yes? So that, when I presented this findings now 10-some years ago, a famous immunologist by the name Pippa Merrick asked me the following question: does differentiation of precursors into regulatory T cells by itself protects against their own self-reactivity? In other words, if all the criminals become law enforcement officers, you deal with the crime. And that means that at one day, if you eliminate all law enforcement officers, there won't be any crime, because all the criminals became law enforcement officers. But it's also possible that only some people with high energy become law enforcement officers. And if law enforcement officers are eliminated, there will be enough criminals left, and there will be crime. So the question was whether the lack of gene, that is FOXP3, equals the lack of cells that expresses FOXP3. Is that clear? So to address this experiment, to address this question, sorry, John Kim, former postdoctoral fellow in the lab, did the following experiment. He generated mice when he inserted into FOXP3 gene a sequence that codes receptor for diphtheria toxin. So in this normal mice, all regulatory T cells express receptor for diphtheria toxin. And its equivalent of this molecule in mice doesn't bind diphtheria toxin efficiently. So if you administer this mice with diphtheria toxin, you can eliminate all regulatory T cells essentially overnight. And this experiment was resulted in striking, so the striking result is shown on the slide. When John eliminated regulatory T cells in perfectly healthy adult mice, mice die from violent autoimmune inflammatory disease within 10 days. So that means that regulatory T cells are essentially guardians for life, and they are essential, they play essential role, and there is essential role for this negative feedback regulation in trans. So life of higher organisms would not continue without this mechanism. So we observed that this autoimmunity happened not only in mice that are colonized by normal microbial community, but also in mice that were made germ-free. Because you can argue that the whole problem of this non-redundant mechanism is because there are lots of commensal microbes and environmental bacteria and viruses and fungi that continuously stimulate the immune system and provide ligands for innate immune sensors. So we made these mice that I described to you germ-free, so they're completely free of any microorganisms. And we found that ablation of regulatory T cells in these germ-free mice results in similar autoimmune inflammation. And microbiota is the largest source of non-self antigens and ligands for innate immune sensors, suggesting or consistent with the idea that it's self-MHC restricted recognition of antigens by T cells, the self-referential nature of T cell recognition, is the cause for the need of this novel and non-redundant mechanism of negative regulation. Now I would like to, how much time do I have left? Okay. All right. Would like to briefly discuss how our studies of the FOXP3 gene illuminated some of the aspects of biology of regulatory T cells. So we found through reverse genetics by different genetic tricks that FOXP3 gene is required in normal cells for suppressor function and fitness, and that the loss of FOXP3 results in a loss of cell identity. So genetic fate mapping experiments that you can do in the lab showed that expression of FOXP3 is stable in differentiated regulatory T cells under physiological and inflammatory condition, but this acquisition of stability occurs in a time-dependent manner, suggesting that there must be a mechanism that allows the cells to remain themselves, to maintain their identity over their lifespan. And we got an insight into addressing this mechanism, another mechanism functions through analyzing regulation of FOXP3 gene expression in vivo. And we focused on so-called conserved non-coding sequences. That's what people thought to be junk DNA, non-coding DNA that doesn't code protein, that involves in regulation of gene expression. And we found that this particular cis regulatory element we call CNS1 is essential for extrathymic differentiation of regulatory T cells. This particular element is essential for heritable maintenance of cell fate during division of differentiated cells. And this element induces, helps FOXP3 gene to be expressed in precursor cells and facilitates their differentiation. I would like to very briefly just show you some of the exciting facets of our studies of these two elements. So we found that mice lacking CNS1 element and impaired in extrathymic differentiation of regulatory T cells were actually surprisingly normal until they reached 6 months of age, and this is sort of my age in mouse years, or months, I would say. So that made us think of what kind of conflict may give rise to this different mechanism of differentiation of regulatory T cells that would be dependent on this particular regulatory element, and we thought of maternal-fetal allogeneic conflict. And when we looked at the genomes of vertebrates, we found that this particular element, CNS1, was present only in placental mammals. And the reason it was, and this is shown on this heat map, where yellow shows 100% identity. So only, and blue is lack of any similarity. So essentially, how one can imagine that one piece of DNA would emerge overnight, or kind of in evolutionary term. We found that much of this gene is coded by retrotransposable element, by DNA that comes from an ancient retrovirus that was jumping and moving around the genomes at the time of radiation of placentals. And this particular element brought into FOXP3 locus binding sites for key transcription factors that are involved in differentiation of extrathymic regulatory T cells. And with this, Robbie Samstein, former MD PhD student in the lab, showed that allogeneic pregnancy is impaired in mice with deficiency in differentiation of extrathymic regulatory T cells, and this is shown here when you see these are embryos and you can count them, and this is uterus of the mouse. And actually the embryos that were born to term showed the features that are typical of the major complication of human pregnancy known as preeclampsia, faulty remodeling of the main blood vessels delivering blood to the fetus. So we speculate that during evolution, mechanism of extrathymic differentiation of regulatory T cells emerged as a result of insertion of this retrotransposable element known as MIR, mammalian interspersed repeat element, into FOXP3 locus to enforce maternal-fetal tolerance. But as frequently is happening in evolution, evolutionary acquisitions are being used for more than initial use that they invented for. And we and others have found that extrathymic regulatory T cells control inflammation at mucosal sites, and we recently found they control features of commensal microbiota, where gut microbial community provides not only antigens to potentiate extrathymic differentiation of regulatory T cells, but also microbial metabolites. And Nick Arpaia, a postdoctoral fellow in the lab, showed that short-chain fatty acid at a site, it's a side product of complex sugar catabolism by bacteria can potentiate extrathymic differentiation of regulatory T cells, and Hiroshi Ono and Wendy Garrett found similar data. And recent unpublished work from Clarissa Menezes and Stas Dickey, two graduate students in the lab, showed that regulatory T cells in turn would affect the composition of microbial community and production of metabolites by microbial community. So CNS2, I told you, maintains regulatory T cell identity when interleukin-2, the key cytokine promoting differentiation of regulatory T cells, is limiting, and when pro-inflammatory cytokines that promote differentiation of effector pro-inflammatory cells are abundant. So in the CNS2 allows for regulatory T cells to maintain their identity under inflammatory conditions, and the one key inducing signal is limiting. And this slide just shows loss of FOXP3 expression, and by now you maybe are used to this flow cytometric plots of protein expression. But it's probably more for scientists in this audience. So when is it important? This is important in situation of chronic inflammation, in all settings of chronic inflammation. CNS2-dependent maintenance of identity of regulatory T cells is essential to control age-related spontaneous inflammation on lymphoid organs, metabolic inflammation associated with either diet-induced or genetic obesity, organ-specific autoimmunity, such as a model of multiple sclerosis in mice, chronic viral infection, and solid organ cancers. So now, what are the signals that are required for these diverse functions of regulatory T cells? We found that in fully differentiated regulatory T cells, T cell receptor signals are required for suppressor function. T cell receptor signals are required for differentiation, but also continuous signaling through T cell receptor is required for the function of regulatory T cells. And we recently found that continuous signaling through interleukin-2 receptor and capture of interleukin-2 by regulatory T cells is required for suppressor function, and regulatory T cells are involved in what you would call social interactions or competition for common goods, competing for interleukin-2 with cytotoxic CD8 T cells. So this is all good, but it seems that there are other aspects of biology of regulatory T cells that keep surprising us. And recently, Nick Arpai in the lab found T cell receptor independent, innate-like function of effector differentiated regulatory T cells and tissue maintenance. What I told you is that T cell receptor stimulation potentiates suppressor function. But what Nick found that in face of tissue damage and inflammation and in cancer, together with Jesse Green, they found in their unpublished work that this result in activation of regulatory T cells and production of factors involved in tissue repair. And this occurs in T cell receptor independent manner and is triggered by necrotic or violent cell death, unintended cell death, or inflammation with two key mediators capable of activating production of immune repair, tissue repair factors by regulatory T cells in T cell receptor independent manner. So Shimon, Dr. Sakaguchi reviewed for you a role of regulatory T cells and therapeutic targets in different settings. So I won't elaborate, but there are a variety of applications of regulatory T cells in cancer and beyond. We ventured at identification of potential markers of regulatory T cells present in human breast cancer as compared to normal breast tissue. And we found several, although we found that the cells have very comparable, similar features in many ways in normal breast tissue and in breast cancer, we found several genes, including chemokine receptor CCR8, cell surface molecule CD177, to be differentially expressed. Is it any useful? We think it is, because we found that in mouse model, punctual depletion, punctual ablation of regulatory T cells can deter growth of primary, highly aggressive breast tumors in mice and metastasis. And these are the tumors that are resistant to currently popular checkpoint blockade inhibition by CTLA-4 blocking antibodies or PD-1 blocking antibodies. So we think that it's possible, and this punctual ablation was transient, so it didn't result in major collateral damage that I showed to you. And also we are pursuing adoptive cell therapy when we can make cells that are expressing active form of transcription factor STAT5. And this is transcription factor activated by cytokine interleukin-2. The cells think that they have seen plentiful interleukin-2. They survive well. They accumulate in non-lymphoid organs. They are independent on interleukin-2, and they have heightened suppressor capacity. And just as the last snippet of data to show you, this is disease observed in mouse model of multiple sclerosis, and few cells that we can, regulatory T cells, where we can make them super suppressive, expand and control disease in this mice. So I would like to close by reminding people in the audience who know, and by telling people in the audience who don't know, that this phenomenon of regulation of immune responses, inflammation by regulatory T cells was termed by Nicole Lidoren and Antonio Coutinho as dominant tolerance. And it's very appropriate, I think, at this time in our history to recognize dominant tolerance. And it's very symbolic, I think, that today we are celebrating phenomenon of dominant tolerance at the time of infectious ignorance and infections intolerance dominating large parts of the world and taking over large countries, including 2 countries I'm coming from. And I hope seeing here high school students, undergraduate students, you are the future, and I hope the excitement of science and excitement about knowledge will carry forward us as a society, as human society, and we will overcome the hard times we are living through. I would like to finish by acknowledging great many former members of the lab working on various aspects of regulatory T-cell biology over the years, and the current lab members, and my many collaborators and colleagues, and Dr. Shimon Sakaguchi for starting the field, and Fred Ramsdell for inspiration and friendship, and thank you so much, all of you. Thank you for your attention.